One day in 1883, at the Graz Zoological Institute in Austria, several scientists were conducting research on marine organisms. They discovered that one of their aquariums had been housing a new species. It lived on the glass wall and measured 3 millimeters, having the smallest measured amount of DNA found in any organism thus far. They named it Trichoplax adherens.
Just two species have ever been described. First, Trichoplax adherens, and second, Treptoplax reptans. The latter has not been seen since it was first described in 1896, and there are doubts about its existence. However, Trichoplax adherens, otherwise known as Placozoa, has been reported in waters taken from tropical and semi-tropical habitats worldwide.
Placozoa are a predominantly asexual organism undergoing budding and binary fission.
However, they can undergo alternating phases of sexual and asexual reproduction dependent on the seasons. Sexual reproduction is also a factor of conditions of extreme warmth or population density, or during a lack of food.
Sexual reproduction occurs when the degenerating body of the mother develops a single egg and sperm, which unite to form a cell capable of division. Initial division within the first cell continues 64 times until it stops and only the DNA continues to replicate. The DNA continues dividing until the nucleus bursts and an adult with 6 cell types is developed.
Due to their lack of muscle, Placozoa use ciliary action in order to move around their environments. Ciliary action, or ciliary gliding, involves changing the shape of the organism in reaction to external stimuli in the environment. Smaller Placozoa are also believed to be able to swim. Here you can clearly see a Placozoa responding to a food source by gliding over to it and eventually engulfing it. Placozoa feed mostly on chromists and algae using a process called phagocytosis. Once it has sensed food, the Placozoa comes to lay on top of the source. Then, it secretes digestive enzymes produced in the glandular cells, through its cells, and over the other organism or food source. They utilize extracellular digestion, and their ventral surface is seen as homologous to their endoderm.
The Placozoa is most closely related to Porifera according to both of our trees. But other than that, it is not very closely related to the other organisms based on features from the data matrix or genetic sequences. The main difference between the two trees is that in the data matrix tree, you can see the development of gills marked by the split from the sea cucumber on. On the other hand, it appears that gills were developed convergently both when the chordata splits from the starfish and when the annelida splits from the mollusca and arthropoda. We believe that these differences can be attributed to the fact that genetic sequences can have slight variations between two organisms, but can cause the expression of completely different traits, placing them further apart on the data matrix tree.
The tree that we developed in lab is different from the tree that we discovered in our journal article, mainly because the article does not use all of the taxa that we did in our tree. For example, Porifera was not placed on the tree from the article where in both of our trees it was the most closely related organism to Placozoa, making it difficult to compare our results with those of the article.
No fossil records have been discovered of the Placozoa. However, the species likely originated very soon after the transition of organisms from unicellular to multicellular.
To date, scientists do not know of any scientific contributions or detriments for humans. While several scientists have hypothesized as to its defense against natural predators like sea snails, their results are inconclusive as to its beneficiality for humans or other organisms.